Yeah, so we're here at Biologists at 100. We have Helena Crellin from the Clare Buckley lab, and you currently have a preprint out on using optogenetics in zebrafish. Is that right?
Yeah.
Can you tell us the story of that preprint in about 1 minute?
Yes. Yes, so our preprint aims to explore how mechanical forces on the cellular level are transmitted to the tissue level in an in vivo vertebrate model. So to do that, we developed this light-activated or optogenetic system that enables us to reversibly manipulate contractile proteins in the cell, specifically actomyosin. And we do that in living zebrafish embryos. And we can do that with high spatial temporal control and at depth. And we were really excited by that technical advance because it let us look at how forces are propagated through complex tissues during development. And we found these surprising patterns in the response of the tissue to induce contractility, which suggested that small local changes in contractility could have an impact over a large distance. And it also suggested that there may be an interplay between mechanical and genetically encoded patterns. So that's a really exciting avenue of further research for us.
Oh, that sounds super cool. So in getting your nice new tools out and your interest in new patterns, what did you find to be the most challenging aspect to then getting to this all to a preprint stage in publishing?
Yeah, so I would say the most challenging part was developing this optogenetic system. So as you might have experienced in your own PhD, that optogenetic, getting optogenetic activation to be reproducible can be really difficult. Yeah. So we developed this light patterning microscope in collaboration with lots of people at the Cambridge Advanced Imaging Centre. And that requires not just customization of the hardware to enable this inverse light patterning, which gives us the ability to have the high spatial temporal control, but also a lot of software customization, so we can have these quite complex imaging workflows. Yeah. Yeah, and then on the biological side, as I've said, the optogenetics can be quite finicky, so that required a lot of optimization to get us to have be able to do these experiments reproducibly.
Yeah, I think it can be quite tricky. So you're here with your poster for your preprint, and what's the most useful feedback you'll either received or you hope to receive whilst you're here at the conference on your preprint?
Yeah, so I haven't yet received feedback directly at the conference, but in general, from the preprint that we've, it's been really helpful to get opinions on our interpretations of the results, as well as we've had some really useful suggestions on experiments for how to further understand these patterns in force propagation that we see. Yeah, I hope that, yeah, basically because the work is very interdisciplinary, getting feedback from lots of scientists of different backgrounds is very helpful for us, because there's always ideas and angles and analysis that we haven't even thought of. So I really hope that means that we'll strengthen the final publication, and I hope that at the conference that I'll get some good feedback as well.
Oh, fingers crossed.
Yeah.
So you talked there a bit about how the preprint may benefit with the research project itself, but how do you envision having a preprint may benefit you personally in your career and your aspirations?
Yeah, so I think the preprint's already helped in my career because I'm in my final year and I'm still writing up my thesis. So the preparation of the preprint's already been helpful for the preparation of my thesis. And then as I'm soon to be in the job market, I hope the preprint will be a good conversation starter with PIs when looking for postdocs. So yeah.
Oh, so you're on the job market. That's good to put out there.
Yeah.
So
gotta let them know.
Oh yeah, of course. And with preprints in general, do you have any general thoughts on the sort of importance of having preprints in the publication system? What do you think is the benefits to having this?
Yeah, so I think there's a lot of benefits to the scientific community. Yeah, like on the personal level, the preprint is a super accessible way to like sort of start publishing your work. And because of the feedback that you get, it helps strengthen your final publication. And then as a reader, it's very interesting because you get to hear about the updates in your field, the cutting edge updates. And on the community level, then I know that it enables scientists who don't have access to paid journals be able to see a version of the paper if it's not then published in an open access journal. So that really just benefits scientific progress.
Which is much more open.
Yeah.
Okay, well, thank you very much for coming to agree to do an interview. We'll put the link to your preprint in the comments. And yeah, I hope the publication process from now on goes smoothly, and that you do find that job from having had your preprint out there. That would be amazing.
Thank you so much.